Hi everyone, welcome to my presentation. Here is Negin Malekian, a PhD student at TU Dagestan. Today I will show you how identification of genes under positive selection in E. coli provided us with a better understanding of targets of positive selection in E. coli and in specific antibiotic resistance. Bacterial phenotype are usually shaped by a strong positive selection. Antibiotic resistance, which is a global health threat, is one of the best examples of such phenotypes. Therefore, we would like to have a deep understanding of targets of positive selection in E. coli and see if positive selection in E. coli targets the genes that are involved in the resistance mechanism. Here, we look into 92 assembled sequences of E. coli, which are collected by our group. To find the genes under positive selection, we perform the DN to DS ratio test, which is the ratio of number of non-synonymous mutation to the number of synonymous mutations inside the genes using the PAML program. So first, we collected 92 wastewater E. coli samples. We sequenced and annotated the genes inside the genomes, and then we filtered the genes that are under recombination because recombination of genes lead to false positives for positive selection test, this kind of DN to DS ratio test. And after applying the Bonferroni correction, we ended up with 75 genes under positive selection. Now, let's look at the function of these 75 genes. As you can see in the first table, the targets of positive selection are diverse. So, for example, they are targeting antibiotic resistance, cellular response to stress, DNA damage repair, and so on. Among this function, we are more interested in antibiotic resistance because it is a global health threat. So, to zoom in more in antibiotic resistance mechanism and the genes with high evidence of positive selection that are involved in this mechanism, we provided you with another table. In this table, you can see how exactly the genes that are involved in antibiotic resistance, they confer resistance. They confer resistance to bacteria. For example, some genes like YFL or YFL, YCGB, and so on are involved in biofilm formation. And biofilm formation is a known mechanism for bacteria to develop antibiotic resistance. Some other important genes are OMPC and OMPR. These genes are involved in efflux pumps, and they lead to reduction of observation of antibiotic by the cell. And also we made a comparison to previous study. So as you can see in the Venn diagram, we compare our studies in terms of number of genes under positive selection with previous studies. And we found that we have some important genes in common, like OMPC and OMPR, which are also, as I said, involved in antibiotic resistance. And in total, we can see that we found more genes under positive selection, more likely because we had more number of samples. In their study, they had around, I guess, 6 or 7 samples, but here we have 92 samples. So that is why we found more genes under positive selection. And also, we were curious to see whether the genes in our study belong to core genome or pangenome. Interestingly, we found that most of the genes under positive selection in our study belong to core genome. And we define core genome as the genes that are present in at least 95% of samples. So, to sum up, we provided a deep understanding of the targets of positive selection in E. coli, and our results show that the targets of positive selection in E. coli are diverse, and among them, we found important genes which are involved in the antibiotic resistance of bacteria. Thank you.